announcements. Upcoming grand rounds on May 15th will be a 2-hour presentation on managing pain, comprehensive care beyond opioids. That would be Dr. Bowen, Dr. Stiles, and Sarah Martin. On May 22nd will be the G5 educational course presentations. On the 29th, the G4 presentations. A G4 presentation by Lawrence Smith. And on June 5th, there'll be a G4 presentation by Michael Baer. For residents and any interested faculty, for those residents who attended the Tuesday Introduction to Quality Improvement lecture, the QI project ideas were due yesterday. If you have not yet submitted one into the spreadsheet, please do so today. All residents have been asked to propose a QI project idea over the next 2 weeks. This is to promote resident engagement and leadership during the COVID pandemic when groups of residents are studying from home. There's no core curriculum today. So it's really an honor to introduce Dr. Jenkins, our speaker today. He's going to speak on the topic of how the Center for Immunology got involved in the SARS antibody testing. Dr. Jenkins and his research team investigate T and B cell activation and tracking of antigen-specific cells. The goal of that research is basic understanding of lymphocyte activation that can be used to improve vaccines and prevent autoimmunity. He's the Regents and Distinguished McKnight University Professor in the Department of Microbiology and Immunology. He's the director of our Center for Immunology. He's this year's recipient of the American Association of Immunologists Lifetime Achievement Award. And perhaps most importantly, he was recently inducted into the National Academy of Sciences, which is really the highest honor in basic research. And this is the first individual since 1970 in the university who's achieved that honor. So, Mark, thank you for being here, but also thank you to your team for the work they've done and provided a great example of linking the value of a medical school and university in that care delivery and basic discovery really are intimately linked in the type of work we're going to talk about, which is critical for our economy moving forward and critical for the medical industry moving forward. So thank you, Mark.
Thank you, Dennis, for that, that kind introduction. And I'd like to thank at least a couple of members of your faculty who've done orthopedic surgeries on me from bike accidents, from which I've had good recovery. So thank you all for that. Okay, so I hope you all can see my screen. And if I don't hear any yelling about that, I'll assume you can. So I'm going to tell, okay, thank you. I'm going to give this talk in the kind of the form of a story, like how the, some of the events that led up to the Center for Immunology getting involved in SARS-CoV-2 antibody testing, give you a little bit of background information about the primary immune response that leads to the production of antibodies, and talk about how the test was developed, and at the end talk about how the test is now being delivered and how it's being interpreted and used in the clinic. And I'll try to do that in about 40 minutes, so there's time for your questions and some discussion. So this is, I must say I don't really give many talks to clinical audiences, so I've never had to really show this before, but I guess I'm embarrassed I don't have anything to disclose. Yeah, we usually work on mice, and I guess I don't usually have to do this kind of thing. So anyway, I thought I would show this email, which is the beginning of my involvement in this process. So this email it was sent to Jacob Tolar, Dean of the Medical School, on March 20th. And this was after a day of many emails being exchanged between people at the university here involved in clinical testing and me as the director of the Center for Immunology. And this was about the time it became very clear that this, the SARS-CoV-2 virus, which was causing the COVID-19 epidemic, was going to be hitting Minnesota, was going to be hitting us pretty hard, and that there were going to be major implications for ongoing research and clinical practice at the University of Minnesota. So, and this was at a time when the molecular test for the virus, a PCR test that detects viral nucleic acid, was being developed by the university and around the country. And at a time when it was becoming clear that even though the test was relatively straightforward, a straightforward PCR reaction done in many labs around the world, supply chain bottlenecks were going to really hamper a lot of people's access to doing the test. So there was one key reagent to isolate viral RNA in a way that was safe from a clinical sample that was limiting. And believe it or not, this was crippling the capacity of people to do the test. And so I was consulting with people in the microbiology lab who were doing that, and it became clear to me that serological testing, that is testing for antibodies against the virus, could suffer from the same problems of supply chain bottlenecks. And it occurred to me that this serological testing might be more problematic because the system might want to test many more people than are tested with the molecular test. People who had no symptoms, just to determine how many people have been exposed. And so I thought that the University of Minnesota and the Center for Immunology could get involved here and do something on this front. So I proposed to Jacob that the university develop an in-house test for serum antibodies to SARS-CoV-2. And my vision would be that the test would really be applied to monitor seroprevalence in frontline healthcare workers and other people in the hospital. And as maybe at the end, we can talk about how that, that plan has been expanded to something much, much more expansive, but that we'll get to that at the end. And so I thought we could do this because we had several assets here at the University of Minnesota that would allow us to move pretty quickly on this. One of the most important was that Fang Li, who's in the University of Minnesota Vet School, is a structural biologist who'd been working on a SARS-CoV-2 protein that forms a critical part of the test. And it turns out that at the time of the pandemic, there were probably fewer than 20 bona fide coronavirologists in the country. And so we were lucky to have Fang Li here. And then I was very confident that the Center for Immunology, with our experience in studying antibody production in immune responses, could develop a good test. And so I thought those things together put us in a position where we could develop a test to measure antibodies to the virus. Now, I knew from talking to my colleagues around the country that other companies, universities would be developing similar tests. And but I felt that this problem of supply chain bottlenecks was so pressing that even if there were other good tests developed around the country for this purpose, we wouldn't necessarily here at the University of Minnesota have access in a timely fashion. It was I also had the sense, based on the claims that were made in some of the commercially available kits that were out early for this purpose, I was pretty skeptical right out of the gate that these were accurate tests. And some of that concern has been borne out if you're following this issue in the media. So I pitched this to Jacob, and he said, go for it. And so the idea was the Center for Immunology team would develop the test, then the test would move into Leo Ferk's clinical laboratory to be further refined, validated, and then put into practice. And so that was the plan. So just again, briefly to talk about the assets that we had at University of Minnesota. And really, first and foremost, was that Fang Li was doing structural biology on the coronavirus. And he published a very nice paper in Nature where he showed that the SARS-CoV-2, so SARS-CoV-2 is a beta coronavirus, that its spike protein, which is on the surface of the virus, interacts with a human protein called ACE2 that's expressed on the surface of lung epithelial cells. And so, here's the structure of the SARS-CoV-2 spike receptor binding domain, and I'm going to abbreviate that for the purpose of this talk as the S1-RBD. And so he showed here, here are the contacts between the S1-RBD and ACE2. Now, structural biology, to solve structures by X-ray diffraction, you need lots of protein to do this. So I knew that Fang had very large amounts of protein to solve these structures. So I contacted him and he confirmed that. And he graciously agreed to provide this protein for the antibody test. So that was critical. I also had a team of investigators in the Center for Immunology, led by Catherine Pape, who were experts on B-cell immunology. And so they've been developing tests to measure antibodies for years, very accurate and sensitive tests. So I was confident that our team could get the job done. I also knew Tyler Bold, who is an assistant professor in the Department of Medicine, Infectious Disease Division, who'd been tasked with collecting samples blood samples from people who are hospitalized with SARS-CoV-2 infections. And so I was confident that we would have access to the samples needed to validate the test. And then, unbeknownst to me at the time, but it quickly became apparent that the lab medicine pathology had a rising star in their department named Amy Karger, who was the head of their clinical lab, which they refer to as the Advanced Research and Diagnostic Laboratory, ARDL. And so Amy, turned out, brought a lot of expertise to the table involved in transitioning the test from our basic science laboratory via Tyler's samples into the ARDL. And so as Dennis mentioned, this is really a nice example of collaboration, not only across the medical school, but across the academic health center. Because of course, Fang Li's in the vet school and the rest of us are in the medical school. We're in a basic science group and Amy's running a clinical lab. So I think it's a great example of how people can work together in the HC and get something done, and get something done quickly if in the case of an emergency. So not to, I know it's early and not to put anybody to sleep, but I want to highlight a few issues about how antibodies are made in an immune response. And as far as we can tell, the immune response to SARS-CoV-2 infection is what immunologists call a primary immune response. Because this virus has never really been circulating in the human population, almost everybody is what immunologists call immunologically naive. We have never been exposed before, and our immune systems are naive. So when we think about antibodies in that context, that means that we would have a few B lymphocytes in our bodies. And here's a schematic version of a lymph node where primary immune responses often begin. We would have a few B cells in our body that have B cell receptors. These are surface antibodies whose antigen combining sites are specific, let's imagine, for the S1-RBD from SARS-CoV-2. These B cells probably exist at a frequency in our body, so somewhere around 1 in a million to 1 in 100,000 of our B cells. So in a naive person, these cells are very rare. But during infection, when the SARS-CoV-2 virus comes into the body, this B cell could recognize the S1-RBD, and that causes it to migrate to a part of the lymph node where it can interact with so-called helper T cells that give it signals to now begin dividing. So this now S1RBD specific B cell starts replicating. And in the process of replication, these B cells then undergo differentiation and become short-lived plasma cells or plasmablasts. And these B cells begin to secrete their surface antibody. And the first form that they secrete is IgM, because the surface B cell receptor is an IgM molecule. So the B cells first secrete IgM, and then they undergo a process called isotype switching. And they switch then the stalk part of their antibody to IgG or IgA or IgE, so-called isotype switching. So in the primary immune response, one would expect to first detect IgM and then other forms of Ig, for example, IgG. So at the same time that is happening, some of the progeny of these proliferating B cells enter what are called germinal centers. And in this germinal center reaction, these B cells proliferate, continue to interact with these helper T cells, and start to introduce mutations into the antigen combining sites of their antibodies. And some of those B cells then generate an improved affinity for the antigen, again in our example here, the S1RBD from SARS-CoV-2, and then leave the germinal center to become long-lived plasma cells. And for many infections, these cells, which reside in the bone marrow, will secrete their antibody for the lifetime of the host. So many of you now have antibodies in your blood right now from your chickenpox infection that you got when you were a kid. And so these long-lived plasma cells are the source of long-lived serological memory, immunological memory. These cells are very short-lived. They peak about 1 to 2 weeks after the infection and then disappear. So early antibodies come from this source, and late antibodies come from this source. So, and then also from the germinal center come some B cells called memory B cells that retain their antibody on their surface. And so if an individual is exposed to the infection, these cells will proliferate, rapidly generate plasmablasts, and increase the amount of antibody in the body. So that's how antibodies get made in response to any primary infection. And so again, in our example, we're thinking about the S1RBD. So how do you measure? So anyone who gets an infection presumably will make antibodies, and we can detect their antibodies in their blood. And because these long-lived plasma cells, in theory, live in your body forever, this is a signature of having been infected in the past, even after the infection is cleared from the body. So antibodies are very useful signatures, biomarkers, if you will, for that reason. So how do you measure antibodies? And so there may be clinical tests done in your field that are based on so-called ELISAs, enzyme-linked immunosorbent assays. And so the system that we developed to measure S1RBD antibodies is done in a two-step format. So the first test is a screening test, which is done on a 1 to 50 dilution of serum from the subject. So, and the way it works is 96-well plates are coated with the antigen of interest. In our case, the S1RBD from SARS-CoV-2, which is provided in pure form by Fang Li's laboratory. So this antigen is absorbed to the plate. Then the free antigen is washed off. Then patient serum at a 1 to 50 dilution is added to the plate. Now, if there are antibodies specific for the S1RBD present, they will bind. Then that serum is washed out. Then we add an antibody made in a goat that is specific for human immunoglobulin, human antibody of all types, IgA, IgM, IgG, IgE, and so on. And that antibody is conjugated to an enzyme called horseradish peroxidase. We then add a substrate that in the presence of horseradish peroxidase is changed from a colorless state to a colored state. And so we can measure this color change. And the color change is proportional to the amount of patient antibody bound to the S1-RBD. So that's the screening test. So anyone who gets a positive screening test then gets a second test. And in that test, the serum is diluted in a twofold series, beginning at 1 to 50, out to like 1 to 50,000. And then antibodies that are specific for either human IgM, IgG, or IgA that are HRP conjugated are added, and then substrate. So this allows a quantitative measure of how much antibody is in the patient's sample. So this test reads out as positive or negative. This test reads out as a titer, which would be something like 1 into 50,000, which meant the serum could be diluted 50,000-fold before the signal disappeared. So the more you can dilute out the sample and retain the signal, the more antibody is present. So that's the system that we developed. And so, you know, to develop the test, we needed samples from patients. And that took a while to get the IRB issues figured out there. That's still, frankly, a challenge for doing this kind of research. I know the IRB does very important work, but in emergency mode, you know, that created some challenges, but that got worked out. And these are the first 2 samples that we actually got from the clinical laboratory. And we were using as controls samples from Hank Belfer's lab provided to us, which were samples from people who had Epstein-Barr virus infections. And these, so this is another viral infection. It's not SARS-CoV-2. And these samples were collected before the pandemic. So these samples could not have antibodies specific for SARS-CoV-2. And so we use these as negative controls. And so here's an experiment where we were coating ELISA plates with either 1 microgram of S1-RBD or 2 micrograms of S1-RBD. And then we were diluting the serum 1 to 50, 1 to 200, 1 to 800. And then doing that ELISA, the screening version of the test, if you will. And then this is a measure of that color change, the optical density. And so what you can see is that the 2 patient samples are the 2 controls from the EBV patients gave very low signal, and we got a positive signal, 1 very positive from 1 person and 1 positive, but less positive from the other person. And both of these people were they had a PCR positive SARS-CoV-2 infection. So we know they were infected. They had viral RNA in their bodies, and they had a hospital grade infection. They were sick. And it turns out that this person was a 38-year-old person who, you know, a relatively young person for this infection. And this was a 69-year-old person who had a lung transplant and was on high-dose immunosuppression. So we thought this was a good sign that we could detect any antibody in this person. So this was, and you can see the 2 microgram coating maybe gives us a little more sensitivity, but not too much. But because we want the test to be sensitive, this is actually the dose of S1-RBD that the clinical lab is using now. So another important question relates to the specificity of the test. And so there are other coronaviruses that can infect humans. And for instance, there are the alpha coronaviruses that can cause common colds. We all have been exposed to alpha coronaviruses in all likelihood, and we all have antibodies. And so we right away, and I remind you that SARS-CoV-2 and its relative MERS-CoV are beta coronaviruses. And so we had a hint right away, in fact, if we go back to this slide, that the test was not picking up antibodies against the alpha coronaviruses, because these EBV infected people almost certainly had alpha coronavirus common colds during their lifetimes, and yet we were getting a very low signal. And you can see that here. And so if we just focus on this, this is that patient who has lots of antibody that we can detect when we use the SARS-CoV-2 S1-RBD on the ELISA plate. If we coat the plate with the MERS-CoV S1-RBD, you can see we lose almost all of our signal. Not all of it, but the vast majority of it. And so the test does not detect the alpha coronaviruses, and it weakly at best would detect antibodies specific for a different beta coronavirus. I would also point out the number of people in the United States who had a bona fide MERS-CoV infection, or for that matter, a SARS-CoV-1 infection from 2002 would number in, maybe 10,000 people at most. So the likelihood of us getting a positive signal from these other infections, even this weak sort of signal, is actually quite low. So then in my lab, we started doing, we were getting clinical samples now from Bethesda Hospital on a more routine basis. And so we started doing the screening test, which the results would get reported as an index value. And so the way that worked is we had a pool of pre-pandemic sera. So basically we took serum from 30 of these EBV infected people and pooled them together to make a pool. And so we would do the ELISA on that pool to get a background value. And then we would test then individual sera from the pool or individual sera from PCR patients from the hospital. And then we would divide the optical density value in the assay from the individual sera by the value from the pool to get what in clinical labs called an index value. And so here are the results we got from the individual samples from the pre-pandemic group. And so you can see they all cluster around an index value of 1, which is what you'd expect because, of course, these are the individual samples that make up the pool. And you can see there are a couple individuals that had slightly more background, but we really didn't see anybody with a value above 2 in the control group. And then these are now samples from patients. And you can see most of the samples give an index value greater than 2, but some of the samples don't. And so we had limited clinical data on the samples we were getting, and it became clear that almost all of the samples that gave a value in the negative range came from people who had had their infection or had had symptoms of their infection for less than 10 days. And so we then plotted the index value against days from symptom onset. And so what you can see here is that all the samples we tested from people who had symptoms for at least 10 days gave a positive result. But then as you walk back in time to earlier points from the time of symptom onset, we start to get the test gets less and less good. And that's really not a good way of saying it, I don't think. I think the test is good. It's just that it takes time, if you remember back to our primer on the primary immune response, for the specific B cells to proliferate, become numerous enough, differentiate, and secrete enough antibody for us to detect. So the test is really good at detecting hospital-grade infections that have, from people who have had symptoms for at least 10 days. Now, epidemiological evidence suggests that most people who get a significant infection develop symptoms about 3 days after their infection. So if you want to, in your mind, kind of guess how, if this axis were to say days after infection, you would add 3 days to each of these values. And so by about 2 weeks after infection, we think the test will detect basically antibodies in everybody who has a hospital grade infection at least, because that's what we've measured. So here are our data now from our basic science lab replotted. And here then is the results of people from my lab working with people in Amy Carter's ARDL lab for them to get the test up and running in their own hands and then validate it to show that they get the same results we get. That's important. And so, and ARDL is really good at this. And so they ran 200 and I think it was about 30 samples from pre-pandemic people. And so they detected a couple that were in this what they call now equivocal range, but really nothing, above now this, index value of about 3. And so this is, again, their testing of some of the same samples we tested, but then additional samples. And again, what you see is basically 100% of the people who've had symptoms for more than 10 days give a positive result in the test. But some people who've had symptoms for less than 10 days don't. And this was another immunosuppressed patient, a different one than we tested, that again gave a negative value. So, that's another limitation of the test. If someone has a lung transplant, is on high dose immunosuppression, which is designed to inhibit the adaptive immune response, that's going to be a problem for tests like this. But in other people, the test is quite good at detecting prior infection. And ARDL, I don't know how many samples now ARDL has tested, hundreds and hundreds. And so this is an example of so everything I showed you was the screening test, which would get reported to a clinician as positive, now equivocal, or negative. So anybody who gets a positive test then gets the endpoint titration test. And so here are samples from 3 patients at different times after their symptom onsets, where we're looking at dilutions of their serum. So the more dilute the serum goes this direction, where we're measuring IgG, IgM, and IgA. And we're titrating that so we get a sense of the quantity. And so this is the negative control samples from negative control pre-pandemic samples. So you can see that, the further out the dilution goes and you still get a signal, the more antibody is. So in every patient at all these times tested, there's more IgG. That's the most abundant isotype of antibody, of S1-RBD antibody in all these people. But they also have some IgM and some IgA above the background. So, so for the endpoint titer, you can imagine sort of draw a line down here at some low value and then extend these lines to ask where would that signal go, cross this very low line, and you get a sense of the titers. So this person has a titer, IgG titer of greater than 1 in 30,000. So that's a lot of antibody. So one issue, if you think back to that early antibody primer, was that you expect early on to have more IgM than IgG, and then as time goes on for IgG to become dominant. And so interestingly, we don't really see that with this infection. So if you plot the amount of IgM that a person has versus the amount of IgG that a person has, there's a very, very good correlation. And some of these samples, especially these samples that are at the low end of the titer range, these are from people who are very early in their infection. So you don't see, values, sitting above the diagonal here for IgM. And so that phenomena has been observed by many other labs now who have developed similar tests. And so that's an interesting immunological phenomena related to this infection. We're not really sure what it means. In everybody who's developed a hospital-grade infection, we also detect IgA, which immunologists attribute special significance to because it's in the mucosal secretions, which might be really important for a respiratory virus. So we also, caught people actually seroconverting. And so these are samples from 3 different hospitalized patients, and we've got their blood at different times after their symptom onsets. And you can see for both IgM and IgG, their antibody titers increasing with time. And so, for a basic immunologist, this is a remarkable thing to observe in a human being, the evolution of their primary immune response over time. You can see some variation in the time it took the people to seroconvert, but as I mentioned, everybody pretty much seroconverts by day 10. And this is, again, really good immunological evidence that the assay is measuring what we think it's measuring. So ARTL is now testing healthcare workers in the mHealth system. And so here's an example of a test that they did. And so they had, in this particular run on that day, they had several samples from PCR positive patients that were greater than 10 days out. And then they had a batch of samples from healthcare workers. And so the vast majority of sera from the healthcare workers were negative. They don't have antibodies, but one person had antibodies. And so you can see there's a really nice separation between the negatives and the positives. And so this person's sample would be called positive, and then would move on to the endpoint titer test. So ARTL now has tested, at the last time I talked to them, which is quite a while ago, so they have hundreds and hundreds of more tests done. The University of Minnesota healthcare worker seropositivity is about 2%, which means 98% of people have not been exposed to the virus at a level that has caused them to make antibodies. So that's a pretty low level. That's being reported in other states that are in early phases of their infection. It's also a testament to the systems that the University of Minnesota has in place that are protecting most of their healthcare workers from the infection. So that's kind of good news, bad news in a way, because the idea now that we're going to have a lot of people in Minnesota who are going to have developed immunity from natural infection is really not borne out here. We're most still immunologically naive. So we've also learned a few things. We have some preliminary information about some of the people who are getting, people like this who didn't go to the hospital, may or may not have symptoms even, we're not really sure. We don't have the clinical data on this person. But so here are examples from 3 people like that. They got a positive score in the screening test. And so here are these 3 people shown here in red, green, and brown. These are their values from the total Ig immunoglobulin test. And here's a sample from a clinical grade infection. So the person with a clinical grade infection has more antibody than these 3 people, healthcare workers who scored as positive. And again, the clinical grade infection is dominated by IgG, although there's IgM and IgA. the people who got these non-clinical grade infections have a range of antibody positivity, but they tend to be less for IgG and IgM. And we can't detect any IgA from these non-clinical grade infections, at least in the small sample so far. Even from this person who had a non-hospital grade infection, but made quite a bit of IgG. So there may be something fundamentally different in the kind of antibody response that people with really bad infections get compared to people with less severe infections in terms of making the IgA version of these antibodies, the kind that goes into secretions. And that has some implications for immunity as well. So, so basically, to conclude about this, and then I'll go into a few kind of implications of this, is that a SARS-CoV-2 S1RBD based ELISA can be used to detect antibodies for the S1RBD. Our data show that all hospitalized PCR positive patients seroconvert by day 10 of symptom onset. That's now consistent with many other similar studies done around the country. S1RBD specific antisera from hospitalized PCR positive patients are dominated by IgG, but also have IgM and IgA. People with non-hospital grade infection may make less S1RBD specific antibody than hospitalized patients, and they may make IgM and IgG, but not IgA. So, you know, that's where we are. The clinical test is now being done basically every day on samples from the mHealth system, as far as I know. So that's where we stand. So I would just mention that you can order the test. I had a fellow from my lab do kind of a mock order. And so that's what these screens are. I don't really know what they mean. I put them up. I assume you will recognize the kind of system that you use to find and order clinical tests. And so you can order them on, you know, your patients if you want to do that. And, you know, this is what the ordered things look like. So I just show that for your information. As far as I know, still the only people who can get the University of Minnesota test, this test, are people who have it ordered by an mHealth physician. So there's this other effort you may have heard of that came from the governor to test basically everybody in Minnesota with the both the molecular test and the antibody test. That's a whole nother kettle of fish maybe that we can discuss. And that system would be, you know, operating in parallel or, you know, independently from this system, which was designed for mHealth workers and patients. So what does a test mean? And so, you know, that's a big issue now. And so this is from the medical school website, and there's still tremendous controversy on this. So I think most people would agree if you have a negative antibody test and a negative test for the virus, there's no evidence you are currently infected or ever were infected. So you are at risk for future infection. So, you know, stay home and distance yourself. So if you have a negative antibody test and a positive PCR test, that is, could and likely means you have an active infection and have not had time to make antibody tests, hadn't made antibodies yet. Definitely, you should be isolating yourself and quarantining yourself at that point. If you're positive for both the antibody test and the PCR test, then you could have an active infection, but later, and again, you should be quarantined. And then there's this category, of course, which is tremendous interest to politicians and policymakers. If you have antibodies but have a negative PCR test, the presumption is maybe you now have immunity and can go back to work. But there are enormous caveats to that idea, given that there are people who, we do not know, number one, are antibodies protective for this infection? Lots of historical data suggests probably. Number two, we don't know if antibodies are protective, we don't know how much antibody is actually needed to get immunological protection. So a lot more work is going to have to be done to figure out what the antibody tests mean. That work is being done. I think we'll have a lot more traction on this soon. But at this point, I think this means people have been exposed and have likely cleared their infection. But whether they should get an immunity certificate, we're way, way too early to be making that call. So that's the end of my presentation, and I guess I'll turn it over to Dennis at this point.
Yeah, Mark, that's just wonderful. Thank you. And for all that you did a great job explaining it to those of us who are not up to date on immunology. So we had almost 120 people on, so that's exciting. There's several questions coming in. I just had one based on what you were just saying. Are there other known diseases where the presence of antibody titers does not confer immunity?
Yes, there are. And there are certain kinds of infections that persist in the face of antibodies. We don't really understand that. Tuberculosis, there are antibodies and the infection persists. But it would be pretty unusual for an acute viral infection like this to have antibodies that don't offer some degree of protection. So there's some anecdotal evidence from SARS-CoV-1 that antibodies were formed and then faded away over years, meaning immunity might fail over time for a coronavirus infection. Too early to tell here.
All right, so our first question is, from Dr. Naas is, there's this concept of the immunologic storm in some patients who seem to be, sicker than others. And is there an explanation for that? Is there an immunological explanation for, the patient who just seems to be clinically so much sicker than other patients and really has multi-organ involvement in some cases?
Yeah, I think there's some understanding of that. Clearly, the magnitude and quality of your immune response is controlled by, we estimate, 1,000 different genes, and people vary a lot in their quality of their immune responses. So there is good evidence that people who make a very robust immune response to the virus make lots of a cytokine called interleukin-6, and that interleukin-6 can cause a lot of the symptoms of this, acute respiratory distress syndrome. And in fact, there are clinical trials now being done to determine if blocking IL-6 will prevent that phenomenon.
All right. A question I had that might be related is, you pointed out that this ACE2 binding site on endothelial cells, it turns out there are some clinical sequelae of the COVID infections that have seemed to be a little bit different. Some patients have had strokes. There's been hypercoagulability. There's been ischemic renal disease. Yeah. Do you think those are connected?
I'm not sure. Yeah, I'm not sure, Dennis. Because a lot of those symptoms were also observed in Ebola virus infection, which doesn't use the ACE2 receptor to enter cells, but has, again, an IL-6 dependent cytokine storm, induces a very, very strong immune response. And a lot of the aspects of potent immunity affect the coagulation system. And so I think those are all related to potent immunity, maybe not necessarily ACE2. That's my guess.
All right, thanks. So a question here, I think we've answered. Can you draw any conclusions about long-term immunity yet? I think the answer is not yet.
Yes, not yet.
And that was from Dr. Bowen. Dr. Van Heest, I think, is asking, as mHealth, this is maybe above your pay grade or parallel to, but not aligned with.
Okay.
As mHealth employees, can we be tested? Maybe I can answer that, Mark.
Okay, Dennis. Yeah, you may know about that, more about that.
I think the answer to the question is yes, you can order the test on yourself. The question that's being debated in the medical school leadership is, should you be tested? And right now, as of Thursday, which I think is yesterday, the answer was you should follow the guidelines. And the guidelines are, if you're tested for this antibody, as Mark said, you should be tested for the virus at the same time. But the guidelines did not cover all healthcare employees. And part of that is a strategy to deploy what are still limited resources to get the most value for our patients and our providers. Certainly a symptomatic worker should be tested. But if we were to test all of our employees, we would not be able to restore care in the ambulatory surgery centers, in our inpatient, facilities, et cetera. So I asked that question to the, Tim Shacker, who's the infectious disease lead on this, as recently as yesterday with Dean Tolar. So that's kind of a long answer, but we'll leave you off the hook on that one, Mark.
Thank you.
And then Mark Tompkins is asking, how did Fang Li's lab obtain the receptor binding domain protein that was specific for the SARS-CoV-2? And do you have any sense of what is the immunity inferred following infection with it?
That's right. Yeah. So after the, of course, the outbreak occurred in China, and the Chinese had sort of they had a bad phase and a good phase, at least as world citizens on this. But in the good phase, they determined the nucleotide sequence of the virus. And that from that nucleotide sequence, you can infer the sequence of all its proteins. And so using recombinant DNA technology, you can now produce individual proteins from the virus. So Fang he cloned the viral nucleic acid into an expression vector and then produced the protein in mammalian cells in tissue culture and then purified the protein. And he did that. There's really straightforward technology to do that. And he did that at huge scale so that, Fang, can provide enough of S1RBD actually for millions of tests. So that is not going to limit our testing capacity. The S1RBD is a very good target for these kinds of tests because it's presumed that if an antibody interacted with S1RBD, it would prevent S1RBD from interacting with ACE2. And so we're working on my lab on confirming that assumption so that I think could inform the clinical debate. So for example, you could have antibodies against S1RBD that bound the side of the protein and would not then interfere with the top where the ACE2 bonds. So we could then report a neutralizing antibody titer that could be more informative. So that is the one thing we're working on.
Yeah, that's a very good explanation and highlights what we still need to know about the various antibodies. So Mark, would then that binding domain protein be used to develop the vaccine, or what are the vaccine strategies?
Yes, there are 8 now clinical trials for vaccines ongoing around the world. There's another 70 that are in process. So every possible viral protein, every possible vaccine modality that's ever been thought of by a human is being tested. But many do target the S1RBD for the reasons I just mentioned. because that is likely to induce neutralizing antibodies.
All right, we have a few more minutes. Dr. Sembrano has a question. If everyone who's had the common cold has antibodies to coronavirus that causes the common cold, I think he means, and yet are not immune to what is different about COVID-19, that may make us think that having antibodies would make us immune?
Well, I think the reality is that the sequence of the proteins that are in the alpha coronaviruses are different enough from the proteins that are in the beta coronaviruses, like SARS-CoV-2. They're different enough so the immunity that we all have to alpha coronaviruses does not cross over into immunity to SARS-CoV-2. In the same way that, our antibodies that we make against the influenza strain from last year may not help us for the strain this year, even though those are related viruses.
All right, we'll take maybe two more questions. They're just pouring in here, which is just really fun to see the level of interest, and it highlights the level of importance of the work for your team, Mark. Is the rapid drop in serum titers typical, or does it suggest there will be no long-term immunity?
Right, that's the million-dollar question. Many labs are pursuing that, and again, that's based on this one paper from SARS-CoV-1. That the antibodies faded over about a 3-year period. It's too early to tell because no one's had a SARS-CoV-2 infection long enough to know whether their antibodies are fading. In the limited set we have, we don't see any evidence for that. That's going to be pursued intensely by many scientists.
Are there other diseases where that's happened, where the antibody titers faded quickly and the disease recurred?
There are, if you might wonder why do you have to get your, tetanus shot, repeated every 10 years, there is some evidence that those antibody titers will fade slowly in some people, but that's over the period of decades, and that's in a subset of people. So for most of the antibody to disappear in 3 years would be basically very few precedents in the immunological literature. I don't think that's going to happen.
All right, I think Dr. Ellerman's question was answered about the neutralizing antibody. I'm just so everyone knows, in fairness, I'm going in order. There are nine new questions. Maybe we have some budding immunologists in the audience.
Yeah.
That would be awesome. We could be on our training grant, and we would connect with Mark and get it all set up. Let's see. Regarding varied immunological responses and antibody test sensitivity, is there a concern for false negative rates in the general population in those with potential history of subclinical infections?
Yes. I have very little, almost no concern about false positives. I have concerns about false negatives in people, as the questioner mentions, who have subclinical infections, probably don't get exposed to much virus. They may have very low level of antibody. So having said that, the test also gives us some information. That person, even if they had an infection, did not have an infection that probably produced immunity. So I think that's a real issue. I think that is going to be the case.
Yeah, thank you. So, Mark, there's a question here from Dr. Morrison. There's been a suggestion of a mutation of the virus and that there's more than one form. And Dr. Morrison's question is the test able to detect the recently reported mutation as well as the original virus? And I guess there's two questions. Are there two forms? And then if there are, is targeting the binding site going to detect more than one form?
Fantastic question. Yes, there are at least two forms there's a mutation in the S1RBD that emerged in China that is reported to be associated with a more pathogenic course of the infection. It's a single amino acid substitution in the S1-RBD. It's not clear what effect that's going to have on the antibody tests. I know that the sequence that's of the S1-RBD that we're using is from the early part of the Wuhan outbreak. That's pretty much what everybody's testing. It's too early to know. My sense is that the test will still pick that up because nobody yet is reporting seronegativity in people with hospital-grade infections of greater than 10 days. So I don't think that's going to be an issue yet. But if the virus does have a capacity to change its genome the way the flu does, then that's going to be that's getting us into the worst case scenario. There's no evidence for that, but that would make it even more challenging.
Great. All right, we're going to be respectful of your time, Mark. I just wanted to thank Joni Bechtold and Patrick Horst, who kind of worked together to contact you. Thank you so much, not just for the presentation, but really the demonstration of the quality of science that's occurring at the university and the value of that, and, which in this case is directly related to not only helping patients, but helping our healthcare workers and our faculty. So thank you for that. Maybe if you're up for it, we'll have you come back in 6 months and give us, part 2 of the lecture of solving some of these mysteries. So thank you so much. And we cannot applaud you here because we're all on mute except for me, but I'm going to do that. So thank you very much.
Thank you.
Thank you for all your